Okay, I think that we can start. Good afternoon, everybody, and thank you for joining to this seminar meeting, this seminar series. It's my pleasure to introduce to our speaker is Professor César Muñoz-Fontela. Professor Muñoz-Fontela is the head of the research group Virology Immunology at the Bernard Notch Institute for Tropical Medicine in Hamburg. His laboratory investigates how the immune system responds against highly pathogenic human viruses, such as those causing hemorrhagic fever. Part of their research activity focus on the development of novel animal models for studying the physiology of this disease, and he is particularly interested in the interaction between viruses and dendritic cells and how this crosstalk impact on the disease outcome and the generation of the immunological memory. Today, Dr. Muñoz-Fontela is going to talk about the epidemiology of Ebola virus disease. So thank you very much, César, for accepting to participate in these seminars, and the floor is yours.
Thank you so much, Jorge, and thanks for the invite. So actually, yeah, as you mentioned, my focus usually is on the immunology of filoviruses, but today I thought that I was going to give a more broader overview of the epidemiology of the filovirus diseases in Africa. And this is also part of the work I do because I also partially work as a consultant for the WHO, and I'm involved in public health policy as well. Can I have the next slide, please? So I put this slide just first to mention that filoviruses are a growing family of viruses, not only Marburg viruses and Ebola viruses, but also other viruses that have been discovered in different types of animals, not only mammals, in fact. And even within the ortho-Ebola virus genus, besides Ebola virus, there's other viruses that are maybe less well known. Some of them are also highly pathogenic for humans, like Sudan virus, Bundibugyo. And then there's others that are really poorly characterized. Reston virus is probably non-pathogenic for humans. Thai forest virus, we really don't know much because there was only one human case in history, and Mobali virus has been recently discovered in different species of bats across sub-Saharan Africa. And within the ortho-Marburg virus genus, we have two viruses, Marburg virus and Raven. Next slide, please. The previous one. Oh, no, so yeah, this one. Thank you, thanks. So this is actually the global distribution of filoviruses across the world. So in green, you see countries that have experienced an Ebola or filovirus outbreak in the past. So as you can see, it's mostly an African problem, sub-Saharan African problem, and it's distributed across Central and Western Africa. You can also see the Philippines in green because of the presence of Reston virus, in particular in monkeys. And then there's other countries where viruses, different filoviruses have been identified, including Tapajos virus in South America. So probably the distribution of filoviruses in the world is greater than we previously thought. And next slide, please. So if you look at the phylogenetic tree of filoviruses, what you can see is that there's a number of viruses that I mentioned that infect mainly mammalian species, including humans, but also non-human primates and some forest antelopes, for example. And there's other viruses that have been discovered in reptiles and also in fish. And you can see that in the phylogenetic tree, that filoviruses like Ebola, Sudan, and Marburg are really, really different viruses. And the reason why I say this is because every time that we have an outbreak of Sudan virus, for example, or even Marburg virus, and we have to contact with the Ministry of Health of the country that is experiencing the outbreak, always they ask us to give them or to bring the Ebola vaccine. We have to explain every single time that the Ebola vaccine actually will never work against Sudan virus, and even less against Marburg virus. So the difference between Sudan virus and Ebola is pretty much the same. The same difference that you see between, for example, measles virus and canine distemper virus. Next slide, please. So filoviruses cause zoonotic infections. That means that they are transmitted naturally between animals and humans, causing disease or not. And so it's necessary that humans are in contact with wildlife in this case to actually get infected. Next slide. So filoviruses are mostly indirect zoonosis. So as opposed, for example, to Lassa virus or rodent-borne viruses that can be transmitted directly between a wild animal and humans, in this case, rodents and humans, or as opposed to all arboviruses that need an arthropod vector to cause infection in humans. Humans can be infected directly through contact with the viral reservoir, which is supposedly bats for most filoviruses, and we know that for Marburg virus is indeed a bat, so people have been infected through direct contact with Rosetus bats. But for example, for Ebola virus, usually you need an intermediate reservoir like non-human primates, and that then transmit the disease into humans. Next slide. So basically, in the ecology of the virus circulates in different species of bats. When I say this, it's always with a lot of caution because, as I mentioned, we know that Rosetus aegyptiacus, or the Egyptian fruit bats, are a reservoir for Marburg virus, but we don't really know which one is the reservoir for Ebola virus. People have identified PCR positive bats or seropositive bats, but nobody has ever isolated infectious Ebola virus from a bat. But assuming that it's in bats, then there's other types of wild animals that are susceptible, and actually they suffer also severe disease, like non-human primates and great apes. And then usually the index case may be a human that can be in contact, maybe a hunter, someone that is in contact with bushmeat, with a dead monkey in a carcass, and then gets infected, and then brings the virus into the village through three very common mechanisms that are very deep in the African culture. One is the traditional funeral practices, where bodies are washed, and there's a lot of contact with the dead body by the relatives. Especially in the first outbreaks, there was a lot of needle stick injuries in unprotected healthcare workers. These days, people are much better prepared, and there's a much better barrier nursing, but still this occurs. And then in the household, where in Western and Central Africa, most people live with extended family. So there's a lot of cases where there are several people within the household are infected. Can you click once, please? And then one thing that has really changed a little bit the paradigm of Ebola outbreaks is that we know now that the filoviruses, at least Ebola virus, can persist for long periods of time in immune-privileged sites of survivors, including, for example, the central nervous system. It can persist in semen, in breast milk, in the vitreous humor. So, and this is a really a game changer because it causes a lot of stigma in survivor communities in Africa, which actually resembles a little bit the early days of HIV in Africa. And people are expelled from their communities, and the communities of survivors have to take care of themselves because a lot of people have been expelled from their families and their communities. And then the other thing, can you click once, please? And then the other thing, which is something that we don't understand in our field, is that in in survivors and in these immune-privileged sites, the virus persists in a genetically stable way. So this is a graph that you see from where you can see the variation in the sequence of a virus that was identified in 2021 in an outbreak of Ebola virus disease in Guinea, and this was isolated from a survivor. And the virus was essentially the same virus that was present in the acute disease in 2014. So the virus can persist genetically frozen, let's say, for a very long period of time. Next slide, please. So which are the spillover risk factors? So there are several of them, but one is the frequency between human-animal contacts, whether the people have pre-existing immunity in the human population that are in endemic areas. Population growth and poverty is a very important factor, and I'll tell you why in a minute. Deforestation, the speed and ease of travel, global climate change that may affect, for example, the migration patterns of migratory bats and others. Can you click once? And this is actually one example. This is a picture of a cave is Python Cave in Kenya, and every year it's full of tourists, and they are just in front of the Rosetus bats where Marburg virus has been detected. Next slide, please. So one specific case is the possible expansion of Marburg virus disease. So we have been recently observing outbreaks in regions that have never reported outbreaks before. For example, in Rwanda, in Ghana, in Equatorial Guinea. It may be because of increased surveillance, it's probably a really big factor, but it can also be that all these factors that I mentioned are resulting in the expansion of the disease. Click once, please. So as I mentioned, the fruit bat, the Rosetus, is a reservoir of Marburg. People have been able to isolate infectious virus from these bats. They are cave-dwelling bats. So growth is directly related with poverty, in particular in the global south. Poverty is related with the deforestation. Poverty is related with illegal mining, which is one of the ways where we know that people have been exposed and infected with Marburg virus. And poverty is also related with capturing bats as bushmeat to increase the intake of protein. Climate change may affect, obviously, deforestation and also the migration patterns of bats. And the only thing that is a little bit disconnected of the factors that I mentioned before is global travel, because this usually happens. So all these diseases usually happen in rural areas of sub-Saharan Africa, and in communities that don't really travel across the world. But this may change, and we have had chances in the past where this may have changed, where we have cases in big cities, in African capitals like Freetown, Conakry, Monrovia, Lagos. And so it's something that could affect, the expansion of a possible epidemic in the future. Next slide, please. So a little bit about the epidemiology in numbers. So as I mentioned, usually outbreaks are amplified by human-to-human transmission. So there's one unique spillover, single spillover event, and then the virus is amplified in the family, in the village, and at the hospital. Next slide, click please. So there's different ways to measure how a virus may spread in the community. This formula is a really simple one where the R naught is a parameter that would let us know if the the virus is going to spread, if the R naught is higher than 1, or if it's not going to spread, if the R naught is lower than 1. So if B is the probability of transmission from an infectious person to a susceptible individual, K is the number of contacts per time unit. For example, how many people do I see in a day? and D is the mean duration of infectiousness, you can have situations that I'm comparing on the right of the slide. In the upper part, you could see the situation of measles in Germany. The B factor is really low because pretty much everyone is infected, everyone is immune against measles virus. So even though I see 20 people per day and the infectiousness is like infectiousness period maybe 4 days, they are not is much lower than 1. You go to the case of, for example, of Ebola virus in Guinea, where no one was immunized, even though that the B is still really low, 0.02, because Ebola is a really infectious virus, but it's not a highly transmissible virus. So you need to really be in contact with infected body fluids. And then they are not is higher than 1. And next slide, please. So how can we suspect that we have a filovirus outbreak, even in settings where you have a lot of diseases that look like filovirus diseases at the beginning, like malaria, typhoid. You have other types of bacterial infections. So if you are in an endemic area and then you have, can you click once? And then you have an infection in one household in the village where people within the household are infected. Click, please. And then you see that the rest of the families in the village are also being infected. That's typical scenario of a filovirus outbreak. As opposed to, for example, a Lassa epidemic, a Lassa outbreak. Click once, please. When you have several families being infected at the same time because the rats go into the houses, for example, in the dry season, and there's infection of several households at the same time. So this is already a big difference that allows us to suspect that we may have a filovirus outbreak as opposed to a Lassa outbreak, for example. Next slide, please. Of course, you can have a different situation where you have an imported case. We had this situation in Spain a few years ago. We had a situation in Nigeria, in South Africa, in the US. So then you need to look at the travel history, contact with wildlife, sick relatives. You need to have a look at the symptomatology, epidemiology, but in particular, you need really good differential diagnosis. Next slide. So in terms of diagnostics, we have there is a huge improvement of the diagnostic methods, I would say, in the last 10, 15 years, where we went from IgM ELISA to virus neutralization, virus isolation, indirect immunofluorescence. That's pretty much what people were doing in the early outbreaks, to PCR, which was the main method of diagnostics in, for example, West Africa, and now there's cartridge-based diagnostics like GeneXperts, where the people that manipulate doesn't really need to be trained, and is really easy. And another point of care rapid tests that are really easy as well. The only problem is that most of this technology has been developed only for Ebola virus and not for the other filovirus. Next slide. So if you look at the history of different filovirus outbreaks in the 80s and the 90s, and then after 2000, there was a change of the epidemic profile after West Africa, because in the early outbreaks, so the outbreak would be in a way self-contained, maybe in one village, 2 villages, sometimes with hundreds of people infected, but it would be self-contained to a specific area in a really relatively short amount of time. Like outbreaks would be resolved in a question of a couple of months. But since West Africa, we've seen that there's an amplification from outbreak to epidemic with more than a country involved, or different areas involved. Or for example, in the recent outbreaks in DRC, we've seen that the outbreak, the epidemic can persist in 2 or 3 areas of the country, but maintain over a year. So there's a lot of discussion these days about which may be the factors that may favor this change from outbreak to epidemic profile. Next slide. So I'm going to show you a video that was done in West Africa. So you see in this map the 3 countries that have sustained the most of the transmission. So in the upper part is Guinea, in blue is Sierra Leone, and in orange is Liberia. And through sequencing, you can see how people with infected with the virus were moving across the 3 different countries. So you see that there's constant movement of infected people across the 3 main countries, with now all the capitals affected, and the capitals acting as hubs for amplification of the disease. And then at the end of the epidemic, you can see some flare-ups that are due to sexual transmission, actually. But you can see that essentially the epidemic was sustained through movement of people. Okay, next slide, please. Click once. So across the main factors that may explain this, one important one is that the 2014-16 West African epidemic was the first filovirus epidemic in history where essentially everyone has had a cell phone. And because of this, because of the improvement in the infrastructures, so that the roads are better and people can travel easier now. So, and in the presence of cell phones, people could call their relatives that maybe were living 80 kilometers or in a different country, and come to the traditional funeral, get infected, and go back to their place of origin. So this is one of the main amplification factors, and one of the reasons why the recent outbreaks in DRC are also maintained over time. Next slide, please. This is a really complicated slide, and unfortunately, you don't see it very well, but I wanted to illustrate the differences between classic interview-based epidemiology and modern molecular epidemiology. So here in the center, you have one case that was identified in Boke. This is one prefecture region in Guinea. It was a young person, a driver that had visited several hospitals, also visited traditional traditional healers, and eventually, by the time that this person was identified and the chain of transmission was identified, he already had passed the virus to his wife, brother, uncle, and neighbor. So, and this was difficult because it was difficult to make the connections between this case in different ongoing transmission chains. Next slide, please. So even though contact tracing and community monitoring is the main way that we can actually prevent the expansion of outbreaks these days, or one of the main ways, of course, in the absence of medical interventions, because it's really essential to understand the change of transmission. So what the contact tracing teams are doing is when they identify a case, they ask the sick individuals to identify contacts, and then the authorities attempt to find and isolate these contacts. You can imagine a lot of situations where this is really difficult. For example, in capital cities, most people move around with taxis, motorbikes, or collective ways of transport where they are in contact with a lot of different people. They are not relatives or not necessarily neighbors. It's a little bit easier to do in rural settings, but still, depending on the also political situation, if you are in an area where there's civil unrest, or even there's war with refugees coming back and forth, it's really complicated, as you can imagine. And then the other thing is community monitoring, where the authorities are looking at the incidence of cases in a particular community daily. So, and then they kind of isolate these communities and limit the travel. Next slide. Yes, thank you. And so, but these interventions are really important. So important that if there's no intervention, so you can see here in the punctuated line, the time of the introduction of contact tracing and community monitoring in an epidemic amplification curve. So if there's no contact tracing, no community monitoring, the reduction of cases is zero. You will see, the green graph, the green line going up, and then you can have reductions depending on how successful you are, from 20% to maybe 60%. Next slide, please. Now, one of the things that happened in West Africa as well was the introduction of molecular epidemiology. This was the first time that sequences of the virus of the samples that we were getting from infected people were sequenced with nanopore technology. And in this graph, you can see the date that the sample was collected and the date that the sample was sequenced. So you can see that sometimes samples were sequenced even on the same day that the diagnostic lab was collecting the samples. And then, can you click, please? So I'm going to show you the same example as I showed you before, a case in Boke. So Boke is the prefecture that you see in brown on the upper left side of the map. Next, click. And so there were cases in 2016 with no known chains of transmission. We didn't know where these cases were coming from. 2016 because there was no transmission at the time. And if you do molecular epi, you can see, you cannot see it very well, unfortunately, apologies for that. But basically, you can see through molecular epi that the case in Boke was a reintroduction of the virus from Koya, so this is the purple area, and that the cases in Siguri, which you can see in pink, was a reintroduction of the virus from Sierra Leone. Next slide. The other thing that molecular epidemiology helped enormously with is to detect and to identify cases of sexual transmission. I will put you one example here. This is a case of a woman, 38-year-old, I think, woman that was infected in 2016. Again, same situation. There was no cases of transmission known at the time, no transmission of the virus in the community, and she was infected. And she passed the virus to her husband and also some of the children. So there was transmission of the virus and casualties within the household. When the teams sequenced the virus, can you click once? Yes. No, one less. Sorry. Thank you. So when the teams sequenced the virus of this woman, the sequence of the virus was essentially the same sequence that was found in the semen of a male survivor that was her neighbor. So imagine how difficult it is to find out this type of situations with classical, interview-based epidemiology. Next slide, please. So besides all these, public health measures, one thing that we've realized over time is that the social engagement and outreach, so the involvement of the community is really essential. And the involvement of the authorities in the community is really, really essential. Giving people the right messages is really important. This is a questionnaire that was given to people in Sierra Leone. And you can see that most people have a high knowledge of Ebola virus disease. So they know that the person with Ebola has a higher chance of survival if he or she goes to the health facility. They know that avoiding traditional funerals is really important. They know that they have to avoid contact with blood and infected body fluids. But yet, at the same time that this questionnaire was given, the persistent drivers of the disease transmission was failure to isolate the sick, unsafe burials, inadequate, for example, sick people seeking treatment for traditional healers. So, giving the message, the right message is really important. At the time this information was collected, there was some messages in the radio where people were informed about the disease, and the authorities would say, Ebola has no cure, therefore if you're infected, you have to go to the treatment center. So people, of course, understood, okay, if Ebola has no cure, why should I go to the treatment center? So the way you convey these messages and the outreach activities that you use to inform people are really, really important. Next slide, please. So the other really important measure that we have for outbreak control these days is vaccination. And there's 2 approved vaccines for Ebola virus disease, only for Ebola virus disease. One is based on BSV, so it's a vector vaccine, and the other is a combination of adeno-26 and MVA. So it requires prime and boost. So as an emergency vaccine, usually the BSV vaccine is the one that is being used. There's other vaccines being developed in the pipeline, including RNA vaccines, inactivated vaccines, attenuated vaccines for different filoviruses. But the only one that we have that is licensed for use in humans is the Ebola virus vaccine. So can you click? Okay, unfortunately, this is not, yeah, now it's good. Thank you. So the way vaccines are tested in the field is always a great matter of discussion with the authorities, public health bodies, stakeholders, vaccine manufacturers, because it's not really ethical, or the ethics committees actually propose that it's not ethical to do a randomized placebo-controlled clinical trial. But one solution that was found is to do ring vaccination, and this is based on the following. Once there's an index case, the people that are in direct contacts of this case and the contacts of the contacts are vaccinated. And that doesn't mean that they have to be relatives. It can be the taxi driver, it can be a friend, it can be someone that has been in contact with a known case. And so these rings are randomized. Can you click next? Thank you. These rings are randomized in immediate vaccination rings and delayed vaccination rings. So the immediate vaccination rings get the vaccine immediately as soon as the index case and the contacts, the index case is diagnosed and the contacts are identified. And then the delayed vaccination rings, in these rings, people are vaccinated 21 days after the establishment of the ring to see differences in the efficacy of the vaccine. This is how the vaccines that we have now, effective for Ebola virus, or as effective as what we have right now, were actually tested in the field. Next slide, please. But there's always, and I wanted to leave you actually with this ethic consideration, because I know there's always discussion about this and it's difficult to figure out. And this is an interview that I gave in a German newspaper, not a newspaper, well, like a magazine, Spiegel, probably, maybe you know it. So where the interviewer were asking me if it was ethical to test a vaccine that was not tested in humans in a clinical trial in the middle of an outbreak, in this case in Marburg outbreak in Rwanda. So my response was, how do you know that the vaccine actually works if you never test it? So we were back and forth, but in this interview they say, why? So in German it says, why should we test untested vaccines in humans? César Muñoz-Fontela. In Rwanda, there's an outbreak of Marburg virus. Since Sunday, investigators test vaccines in people. One virologist, me, says this is not only ethical, this is actually necessary. And so, next slide. So I want to leave you with this, and just again thank you for your attention, and thank you again, Jorge, very much for the invite.
Thank you very much, César, for this great talk. And we have time for questions. So if somebody in the audience want to ask to César something, just open the micro and go ahead. César, I have a question for you. In the meantime, regarding to the vaccine and the efficacy trials, do you think that we need to imagine different way to test or to accelerate this vaccine for this kind of pathogen? I mean, we can use information that we are getting from animal models to accelerate the implementation of this new vaccine for this emerging virus that we don't have too many time to test this vaccine. That there is alternative to the classical or the approach that you show.
Yeah. I mean, there's different approaches. One is immunobridging, and this is actually how the Janssen vaccine was approved. So this is the adeno 26 MVA. So from essentially immunology data that was collected in non-human primates. In the US, there's the animal rule that essentially says that if a vaccine is effective in non-human primates, it's likely to be effective in humans and can be further, go into clinical trials in humans. This is also, I mean, the other part of this ethics is that there's a lot of restrictions and a lot of ethics constraints also to do studies in non-human primates outside the US. And then one thing that is attempted and is kind of in place already is, so WHO has developed a core protocol pathway where protocols to test vaccines are pre-approved by ministries of health of the endemic countries. And the idea is that if you use the same type of approach with the same SOPs and the same protocol, you can get cumulative data from different outbreaks from now into the future, and accelerate, the licensing of vaccines for other viruses rather than Ebola or additional vaccines for Ebola. Because otherwise, what happened in the past is that from each one of the outbreaks, you need to collect as much data, but then you are lost when the next outbreak happens. So the idea is to try to collect cumulative data over the years to accelerate licensing.
Okay, thank you. And what we know about what is the mechanism of protection of this vaccine? I mean, what is the role of antibody? What is the role of T-cell responses in these vaccines?
Yeah, it's actually not really clear, I must say, because one thing that is clear is that the VSV vaccine doesn't, I mean, the protection exerted by this vaccine is based on the presence of antibodies, not necessarily neutralizing antibodies, because antibodies that work through FC receptor functions are also apparently really important for protection. And one thing that we know is that it produces a really limited, I would say, T-cell response that decays over time relatively fast. So I think that we need to understand still the duration of immunity. There's been some suggestions that people that were vaccinated in West Africa were reinfected or could be reinfected. So we don't know how long the protection lasts, and we don't really know very well the correlates of protection, but it's probably based on the antibody response.
Okay, thank you. We have a question in the chat. I think that you can try to open the micro, but in the meantime.
Jorge, yes, sorry, now it works. Sorry,
Okay.
Thank you so much. But it was not working before. That's why I wrote the question in the chat. So César, thank you very much. This is Nuria speaking. Beautiful talk. I was wondering, do you think that the new mRNA vaccination strategies could really become a game changer in the near future? Do you think that they could be implemented to protect against the different filoviruses, or do you think that we are far away from that?
Thank you, Nuria. I think on paper, yes. I think that the mRNA vaccines have the potential to be really effective. I see a problem with cost associated to this type of vaccines. I see a problem associated to production, and I see a problem with cold chain maintenance. I think I see more like economic type of problem rather than science problem. You know, I think that the main companies that may be interested in that will think twice before producing doses of an mRNA vaccine that is really costly in peacetime or in the presence of small outbreaks, you know. And so I think that's the main problem, but potentially yes.
Okay, thank you so much, César.
César, regarding to the reinfection that you commented before, do you know if this reinfection is developing a severe disease, or still they are partially protected even they get infected?
I cannot tell you about reinfection. I don't think we have any specific case of case reports of a reinfected individual. I think this is mostly from epi data from the people that were in the field, but we don't have any specific case that we study. What we know is that people that have persistent infection, right, can relapse with full-blown hemorrhagic fever. So even if the person has had an acute infection, recover, and develop natural immunity, if that person relapse, it may be as severe as the first acute infection. That we know.
Okay, very interesting. And what is the role? What is the role that the innate immune system is playing in the control of the infection, or even in the spread of the virus and causing the disease? Because this virus, I think that they have the capacity to replicate in innate cells. So
yeah.
And to me, something that is important to understand with this kind of virus is the connection between humoral immune responses, the presence of antibodies, or let's say no highly efficient antibody neutralizing antibodies, and also the connection with the innate immune system and the capacity to generate this antibody dependent enhancement. What is happening with this virus in this context?
Yeah, this is really interesting, and that's actually one of the topics that we are studying in the lab, because there's been a disconnection between in vitro data and clinical data. So the in vitro data says that the virus infects primarily macrophages, activated monocytes, and dendritic cells, and because of the expression of interferon antagonist proteins, it can block the activation, and it can lead to, I mean, it was pictured as an immune suppressive disease because of this. But the fact is that people that are infected have huge levels of inflammation and huge levels of T cell activation. So we have seen that, for example, comparing all the dendritic cell subsets that you have, some are kind of suppressed, but some of them are highly activated. And these highly activated ones can activate T cells. So that creates, a link between in vitro and in vivo data. But these days, it looks more as an excess inflammation disease, more really similar to sepsis, for example, where there's a dysregulation of the immune system. I don't know what I was going to say, but yeah, what I was going to say is that this is difficult to figure out in humans, because when people come to the treatment centers, they are already really sick, right? They have high levels of anemia. So probably this immune suppress mechanism exists, but maybe during the incubation period, at early times after infection, where we never get to study patients. But at the end, there's definitely really, really high levels of inflammation.
Okay, thank you. We have a question. Julia, go ahead, please.
Thank you, Jorge. Thank you for the talk, César. This is Julia. I have a question regarding the mechanisms of viral persistence. I don't know if the number of cases is enough to draw any conclusion, but is there any immune correlate of viral persistence?
Yeah, this is a really, really interesting question. So I think that the numbers are enough to make conclusions that a percentage of survivors, especially male survivors, have infectious virus in semen for months or even years after recovery, is enough to say that a number of survivors have post-acute infection sequela, including neurological signs, arthralgia, which may be related with persistence in the central nervous system. What we don't know is the mechanism or any correlate that any biomarker that would inform during acute infection about persistence afterwards, for various reasons. One is because people were not looking at this type of biomarkers, and then because there's no really a correlation between, for example, viral loads or severity of the acute infection and persistence. What is known, and this is a little controversial and has to be studied even more, is that at least in non-human primates, those where persistence virus has been found were those essentially treated with monoclonal antibodies. So one possible mechanism is because of when you treat with monoclonal antibodies, the antibodies take the virus into the monocytes, for example, through Fc receptors, and then the monocytes infiltrate, for example, the central nervous system, where they become microglia in inflammatory conditions. So this is one possible mechanism. So that would mean that through the use of these monoclonal antibodies, we are saving people from dying, but we may be creating persistence in survivors. But I'm speculating a little bit. So, but the literature in non-human primates suggests that that could be one reason.
Okay, thank you, César. I'm following this speculation. Do you think that the vaccine can also work in the same way? I mean, people that get vaccinated, but they are not completely clearance the virus, they can also establish a persistent reservoir without symptomatology?
I think with the I don't think so. I think that with the vaccine it would be different, right? Because you are inducing immunity against the viral antigens, mostly against the glycoprotein. So I think that would prevent the virus. At least the efficacy data suggests that it prevents the virus from replicating to the point where you can get to this problems, no, and even preventing people from getting sick. So I think that this may be more a consequence of manipulating the system where there's high levels of virus. You know, the levels of viremia are unthinkable, and as we were saying before, in the context of really high inflammation. I can tell you that we started a project in the lab and trying to use CAR T cells against the glycoprotein during infection to see if it had a therapeutic effect, and we made things worse. Yeah, in the mouse model. So it's difficult to actually, patients these days as a standard of care receive steroids. So the main idea is actually to lower the inflammation.
Okay. Okay, thank you. More questions from the audience? Regarding, César, very interesting to me, the animal model that you are using for studying this kind of virus. For Ebola, you comment the mouse model, but for other viruses, hemorrhagic virus, is well established the animal model that we can use for studying this disease, these viruses?
Yeah, I mean, the mouse model actually is not very good. I mean, we used it because in our BSL-4 lab, we have the capacity or the knowledge to use non-human primates, and I think also we would never get permit. So we have used in the past mouse models. So filoviruses can infect mice, but they don't cause disease in mice, right? Which is actually really interesting. And you can only cause disease if you have interferon receptor knockout mice or STAT1 knockout mice. So it's not a really good model because it doesn't recapitulate the disease really well. I would say it's really the same with Lassa. Other hemorrhagic fevers, there's not really a really good model for hemorrhagic fevers in mice. So non-human primates are a much better model. Ferrets are being used, and it's not a bad model either. And there's other models where they get sick, but there's not a lot of reagents, you know, like hamsters or guinea pigs. So it's difficult to study in immunology, which is what we do.
Yeah. Okay, thank you. We have. I don't have any question in the chat. More question for César? We have another question. Please go ahead. Open the micro and.
Hello, it was a great talk. I just have a very quick question just about the vaccines. Just how effective are the current vaccines in terms of controlling infection, and how much of like a population would have to be vaccinated for it to actually be effective?
Thank you. So the vaccines are the BSV vaccine has been tested in phase 3 clinical trials. So this was the endpoint was efficacy, vaccine efficacy, and it has been shown that it's an effective vaccine, and even in the delayed rings, it has a protection effect compared to people that did not receive the vaccine. In terms of apologies, I forgot the second question. I'm getting on.
Yeah, it was how much of like the population would have to be vaccinated? Yeah.
Sorry. Yeah, this is actually one of the most difficult things to figure out because we don't know when or where the next outbreak is going to happen, right? So it's to do a vaccination campaign in a specific area, you would need different things. So you would need to have a pharma company, biotech company engaged and involved in producing enough doses for a certain number of people, which in Africa we're talking about huge numbers of people. We would need to select one specific area that we would start vaccination with, and this is really difficult because would you start in the Democratic Republic of Congo, in Guinea, in Uganda? It's really complicated. And then you will need to reach rural areas which have problems like cold chain access, logistics. So it's really a problem. And so the vaccines are being used as an emergency vaccine when the outbreak is detected. And what I can tell you for sure is that they have been very effective at controlling the outbreak, comparing to previous outbreaks where we were talking about Ebola virus disease, in which we did not have vaccines. Also because the vaccination teams and the contact tracing teams, they have so much experience now that they can implement this really fast. But it's difficult to start a vaccination campaign because you would not have enough doses, and you would not know where to start and when.
Thank you, Cesar. More questions? We have a couple of minutes. If not, we can finish here this session. Cesar, thank you very much for your contribution to this seminar series, and thank you to the audience for this great discussion. And we will see you in the next seminar. So enjoy the day. Thank you.
Thank you. Bye.
Bye-bye.
Thank you very much. Bye.